Hello everyone, I'm Xingyue from Duke Kunshan University. Welcome to my bio presentation on cell cycle and observations through microscope. The cell cycle is a sequence of events that a cell undergoes from its formation to its division into two daughter cells. It contains different phases, interphase and mitotic phase, including mitosis and cytokinesis. The cell cycle is a tightly regulated process that ensures the accurate replication and distribution of genetic materials and the proper growth and functioning of the cell. This fundamental biological process is essential for the growth and development of living organisms. Let's go through the different stages of the cell cycle. The interphase is crucial in the cell cycle, representing the period between cell divisions. It is the longest phase and is not a part of the cell division itself. It can be divided into three stages, G1, S, and G2 phases. The G1 phase is the first gap. The cell grows in size and synthesizes RNA, proteins, and other molecules necessary for cellular functions and growth. S phase is the synthesis phase, when DNA synthesis occurs. The cell replicates its DNA to ensure that each daughter cell will receive a complete set of genetic information. Also, the centrosome is replicated in the S phase in the preparation for cell division. In G2 phase, the second gap, the cell continues to synthesize proteins, RNA, and other molecules necessary for its growth and function, and undergoes final preparations for cell division. Together, these stages of interphase prepare the cell for mitosis. Interphase is the longest part of the cell cycle, and the majority of the cell's life is spent in this phase. It ensures that the cell is ready for its accurate transmission of genetic materials during cell division. Mitosis is a process of cell division that ensures the accurate distribution of genetic materials from a parent cell into two identical daughter cells. The mitotic process contains several phases: the prophase, prometaphase, metaphase, anaphase, and telophase. During prophase, the cell gets ready for division. Chromosomes, which are structures containing genetic materials, become visible as they condense. The nuclear envelope, a membrane that surrounds the cell's nucleus, starts to break down, allowing the chromosomes to move freely within the cell. Microtubules from the centrosome start formation. When it comes to prometaphase, the nuclear envelope completes its breakdown. Microtubules attach to the centromere of chromosomes, moving the chromosomes towards the cell center. In metaphase, the chromosomes align at the center of the cell, forming what's known as the metaphase plate. Microtubules are fully formed and connected to each chromatid. This alignment is crucial for ensuring that each daughter cell receives an identical set of chromosomes during division. Anaphase is the separation of sister chromatids. Each chromosome, consisting of two identical chromatids, is pulled towards opposite ends of the cell by the microtubules. This step ensures that each daughter cell receives a complete set of chromosomes. Telophase is the final stage of mitosis. The separated chromatids reach opposite poles of the cell. A new nuclear envelope forms around each set of chromosomes, creating two nuclei within the cell and initiating their decondensation. Cytokinesis is the final stage of the cell cycle and involves the division of the cell's cytoplasm and other organelles into two distinct daughter cells. Our biology lab uses the garlic root cells in the microscope to observe the cell cycle. Here are the materials and the experimental steps. First, cut a few 1 centimeter long roots from actively growing garlic plants. Put these roots in a glass container filled with a fixation solution, aiming at fixing the cell growth for observation. Then, transfer the treated root tips into a glass watch. Introduce 4 to 5 drops of 1 mole of HCl into the watch, covering the root tips completely. Wash the root tips 2 to 3 times with water to remove the HCl. This HCl step is to soften the cell wall in preparation for stained color. Next, apply 2 to 3 drops of gentian violet stain to cover the root tips. Use the water bottle to wash off excess stain 2 to 3 times. For slide preparation, place a single root tip on a clean glass slide and add a drop of distilled water to it. Apply pressure on the cover slip, flattening the root tissue to obtain single-layered cells. Finally, examine the slides under low magnification to locate root tip cells. When finding the cells are clear under low magnification, switch to a higher power objective lens to observe the cellular structure at various developmental stages in a clearer way. From the lab result, we can observe the Gaulic root tip cells at interphase, prophase, metaphase, anaphase, and telophase. Mitosis is important through one's whole life, not only in the most rapid developmental stages in infancy and childhood. Here is one of the real-life connection, the wound healing, which is a central role in the regeneration of damaged tissues. After an injury, cells near the wound site undergo mitotic division to replace lost or damaged cells. This process is triggered by the inflammatory response, which initiates signaling pathways leading to increased cell division. The outer layer of tissues will actively undergo mitosis during wound healing. Overall, mitosis is essential for the timely and coordinated repair of tissues, ensuring the restoration and functionality after injury. Mitosis is also closely connected to the development of tumor and cancer, which is a severe global health issue. Mitosis contributes to tumor development through the disruption of its normally regulated cell division processes. Genetic mutations in key cell cycle regulatory genes lead to uncontrolled mitotic division, initiating the formation of tumors. The rate of cell division within the tumor is increased. Moreover, the unrestrained mitotic activity allows cancer cells to invade surrounding tissues and organs. Recognizing the abnormal mitosis in cancer progression has guided the development of targeted therapies. For example, anti-mitotic drugs aim to disrupt the mitotic process, particularly the formation of the mitotic microtubules that ensure accurate chromosome segregation. By hindering these processes, these drugs aim to inhibit the uncontrolled division of cancer cells and lead to cell death. Here comes to the end of my presentation on cell cycle and observations through microscope. I would like to express my appreciation to Professor Li for his dedication to the Bio 110 course. His support and energy has been invaluable throughout our learning journey. A special thanks goes to our diligent lab coordinator and technician who consistently provided clear instructions and materials for our weekly labs. I would also like to express gratitude for our two amazing tutors, whose comprehensive summaries and explanations in study groups greatly contributed to our understanding of key concepts. Finally, a big thank you to my ThinkPeershare group and lab mates. Your insightful contributions and engaging discussion have helped deepen our collective understanding of biology knowledge. Thank you all for being an important part of my biology journey. Here is the reference websites of the pictures in my presentation. Thank you for your listening.